In the chemical equation of cellular respiration, organic compound glucose is degraded with oxygen present in the reactant and forming carbon dioxide, water, and ATP in the product. Cellular respiration is a catabolic pathway that breaks down complex molecules such as stored energy in nutrients and provide energy usable ATP for metabolic process of life. Glycolysis is the first step of cellular respiration that occurs in the cytosol of the cell. In glycolysis, glucose is the initial fuel of energy force. It contains 6 carbon sugar. 2 ATP is used and release ADP to break down carbon molecule into 2 molecules of 3 carbon molecule called pyruvate. During the process, NAD is used to release NADH, and ADP is also used to release ATP. The output of glycolysis is 2 pyruvate, 2 ATP, and 2 NADH. The second step of cellular respiration is the citric acid cycle, or the Krebs cycle that occurs in the mitochondrial matrix. If oxygen is present, pyruvate is able to transport into the mitochondrial matrix by the transport protein. If oxygen is not present, then it is a fermentation process. When pyruvate is transported into the mitochondrial matrix, pyruvate loses carbon dioxide and coenzyme is added to it to form acetyl-CoA, a 2-carbon molecule. In this process, NAD is used and NADH is produced. Acetyl-CoA is oxidized to form carbon dioxide in the Krebs cycle. NAD is used and NADH is produced. ADP, an inorganic phosphate, is used to produce ATP. FAD is also used and FADH is produced in the process. The output of citric acid cycle is 4 carbon dioxide, 2 ATP, 6 NADH, and 2 FADH. The third step of cellular respiration is the electron transport chain and chemiosmosis that occur in the cristae. NADH and FADH released in glycolysis and citric acid cycle contain high-energy electron extracted from food to an electron transport chain built into the inner mitochondrial membrane. The yellow arrow is the pathway of electron transport. NADH and FADH are used. NAD and FAD are released during the process. The pathway finally passed to 2 hydrogen plus half oxygen at downhill end of the chain, forming water. As electrons pass through the protein complex, they accept and donate electrons and pump hydrogen ions from mitochondrial matrix into the intermembrane space, which create an hydrogen gradient across the membrane. In chemiosmosis, the hydrogen ions flow back down to their gradient through the channel of ATP synthase, another complex protein. The motion of hydrogen gradient flow from high concentration of the inner membrane space to lower concentration into the mitochondrial matrix by ATP synthase. ATP synthase trap phosphorylate ADP to form ATP. The output of electron transport chain and chemiosmosis is 2 NAD to FAD to water and 32 to 34 ATP. The maximum number of ATP that a glucose can produce is about 36 to 38 ATP.